Imagine you're standing in front of, like, the world's largest, most comprehensive library of medical knowledge,
Oh, wow.
right? Every cure, every failed experiment, every tiny chemical tweak made by the brightest minds in pharma over the last 30 years is just sitting right there on the shelves.
That sounds incredible.
Yeah, but then you walk in and you realize there's a catastrophic problem. All the books are written in invisible ink,
Oh, of
and
course.
the pages have been ripped out, they're scattered all across the floor, and the master index was, like, burned decades ago.
So it's basically useless.
Exactly. You know the answers to some of the most complex diseases on Earth are in that room, but you physically cannot read them. So welcome to the Deep Dive.
Glad to be here.
Today, we are exploring a genuinely mind-bending new research paper out of 𝗘𝗏 Ltd. about a system called Harvest. And the mission today is to show you exactly how a new breed of agentic AI just walked into that messy library, turned on a massive blacklight, and started reading.
And reading fast, I might add.
Oh, unbelievably fast. We are talking about unlocking billions of dollars of dark pharmaceutical data. And when I say unlock, I mean this AI just completed the equivalent of 55 years of continuous human expert labor
Yeah.
in under a single week, all for the price of about 11 cents a document.
It's honestly a staggering operational achievement. I mean, we are talking specifically about pharmaceutical patents here.
Right.
And these documents contain vast oceans of experimental drug data that are legally public. Like, anyone listening to this right now could just go download them.
And I'm guessing there's a catch.
A huge one. Because being legally public is very different from being computationally accessible. And what's fascinating here is that this specific data gap isn't just some, boring archiving problem.
Right, it's maybe than that.
It is currently causing a massive generalization crisis in modern AI drug discovery. We've built these incredibly powerful billion-dollar AI models, but they are effectively starving for the very data that is trapped inside these patents.
Okay, let's unpack this. Because before we can understand the actual AI solution, this whole Harvest system, we first need to establish why this data was dark and invisible to human scientists in the first place.
Yeah, it's a good place to start.
Like, if this data is so incredibly valuable, I'm trying to figure out why it's hiding in messy patents instead of sitting in nicely formatted, peer-reviewed academic journals.
Well, the timeline of pharmaceutical development basically dictates it. Pharmaceutical patents are arguably the single largest repositories of protein-ligand interaction data, or PLI data, ever assembled.
Okay.
When a pharma company discovers a potentially groundbreaking new molecule, the very first thing they do is protect it, right? They file a patent years before they ever run clinical trials.
Ah, I see.
And certainly years before they publish a polished peer-reviewed article. And these patents contain what the industry calls structure-activity relationship tables, or SAR tables.
So SAR tables.
Yeah.
I imagine that's essentially the raw recipe alongside, like, a massive spreadsheet of test results proving how well different variations of that recipe work.
That is a great way to visualize it, yeah. These SAR tables document the binding affinities across hundreds, sometimes thousands, of slightly altered compounds. But the problem is how that data is formatted. It's completely unstructured. It's buried in sprawling, complex tables that might span 40 pages.
40 pages of just tables.
Yeah, mixed in with dense legal jargon, and worst of all, chemical diagrams that are embedded purely as image files.
Oh, that sounds like a nightmare for a computer to read.
It is. The current gold standard public repository for trying to collect this kind of data is a database called BindingDB. But BindingDB is severely bottlenecked.
Because humans are doing it.
Exactly. It relies on slow, manual curation by human experts who actually have to sit down and read through these monolithic documents.
And humans just cannot keep up with the sheer volume of a global patent office. I mean, the paper points out that U.S. patents contain an average of 160 distinct experimental measurements per document.
Right.
If you compare that to a standard academic article, which only has about 40, you really see the scale of the problem. Automating this has historically failed because the info is just so fragmented.
Totally fragmented.
But, if human eyes are too slow, we logically turn to scanners. We already have massive public tools like 𝗫𝗫𝗄 that index chemical structures at scale.
Sure.
They act like an automated scanner for the library. So why can't we just point those existing scanners at these patent tables and have them read the numbers for us?
It really comes down to a lack of semantic depth. A single-pass system like 𝗫𝗫𝗄 is fantastic at answering the what.
Okay, the what?
Yeah, it can look at a document and see the chemical structure. It can tell you, hey, this patent definitely mentions molecule X,
Right.
but it completely lacks the relational architecture to systematically extract the quantitative binding values and map them back to the specific biological targets.
So you can't connect the dots.
Exactly. It cannot tell you the context. It can't say molecule X was tested specifically against this lung cancer protein in this exact laboratory assay and achieved this precise binding affinity. Single-pass automation tools just fail to capture that full relational picture.
So we need a fundamentally new architecture. We need a system that doesn't just skim for keywords, but actually reads the text, understands the physical chemistry being described, and mathematically connects the dots across dozens of pages.
Yes, which brings us to the harvest pipeline.
Right, harvest.
Harvest stands for High Throughput Agent Retrieval of Values for Evaluated Small Molecules and Targets. It is a multi-agent large language model pipeline designed specifically to process patent XML files.
Multi-agent.
Yes, and that prefix, the multi-agent part, is the crucial mechanical breakthrough here.
Because if you just hand a 200-page patent to ChatGPT or like a standard single large language model and ask it to pull all the data, it's going to fail, right? I'd imagine the context window just shatters.
It fails miserably. And the researchers tested exactly that. When they tried a monolithic single prompt approach, they hit two major walls.
Okay, what was the first one?
The first is what we call attention dilution. Patent documents can easily exceed 200,000 tokens. When you ask a single LLM to hold all of that context in its working memory simultaneously, it loses the thread.
It just forgets what it's reading.
Basically, yeah. It's the classic needle in a haystack problem, but worse because there are thousands of needles. It forgets which specific compound is associated with which biological target by, like, page 80.
Wow, okay. And the second issue?
The second issue is task interference. If you ask a single model to simultaneously extract complex chemical names, identify biological targets, and pull numeric values, its neural pathways basically get crossed.
Like it gets overwhelmed.
Yeah, it gets confused and actually starts hallucinating fake chemical names that don't even exist in the text.
Here's where it gets really interesting. Because instead of giving one overwhelmed intern a massive stack of 200-page patents and screaming, find everything.
Which never works.
Right. The harvest system builds a highly specialized, rigidly controlled factory assembly line. It uses 5 sequential agents, each with a very narrow, incredibly specific job.
Yes.
And they don't even know what the other agents are doing outside of what is handed to them.
The decomposition of the task is brilliant. Agent 1 goes in first, and its only job is to scan the text for biological targets and the assay conditions.
Meaning the specific laboratory environment the test was run in.
Exactly. It ignores the chemistry, it ignores the math. Once it finds the targets, it passes that isolated context down the line to Agent 2.
Okay.
Agent 2's only job is to extract the quantitative measurements, the 𝗖𝐶50, key, and KD values, and strictly link them to the targets Agent 1 just found.
Okay, let's pause on those acronyms for a second so we're all on the same page. 𝗖𝐶50, key, KDD, in layman's terms, these are essentially measurements of potency, right?
That's right.
Like, they measure exactly how much of this chemical powder you need to sprinkle onto a specific protein to shut it down or alter its behavior.
That is precisely what they measure. It's the physical proof that the drug actually works.
Got it.
Now, after Agent 2 links those numbers to the targets, we hit a translation problem. In patents, lawyers and chemists rarely use the full, terrifyingly long chemical names in the actual data table.
Because that would take up way too much space.
Exactly. They'll just write shorthand, like example 1 or compound 42. So Agent 3 steps in purely as a translator. Its job is to scour the text to map those shorthand aliases back to their actual chemical identifiers.
And then Agents 4 and 5 act as the ultimate standardization checkpoint. Because an LLM's output is just raw text, but a computational chemistry model needs hard mathematical data.
Right. The formats have to match.
So these final agents take those chemical identities and protein names and resolve them into standardized, universally recognized computer formats. Smile strings for the chemicals, and canonical uniprot identifiers for the proteins.
And those SMILES strings are critical here. It stands for Simplified Molecular Input Line Entry System. Right, but it's just a way to take a complex three-dimensional chemical shape with all its rings and branches and translate it into a simple line of text that a computer can instantly read and render.
I want to zero in on a specific technical hurdle regarding those chemical structures because this is a detail that just blew my mind.
Oh, the file conversion issue?
Yes. The researchers found that standardizing the chemical diagrams using existing industry tools was actually actively corrupting the data. How is the standard tool breaking the chemistry?
Well, the root of the problem is file formats. The U.S. Patent Office embeds chemical diagrams into their documents as either MOL files or ChemDraw binary files, which are known as CDX files.
Okay.
The standard practice in the cheminformatics industry is to just blindly convert everything into MOL files so it's uniform. But the researchers discovered that the default conversion algorithms were butchering the translations.
How so?
Chemists often use shorthand in their diagrams, writing ME for a methyl group or ET for an ethyl group.
Right.
The standard converter would occasionally hallucinate during the conversion and misinterpret those shorthand text labels as literal carbon atoms.
Which completely changes the physical shape and weight of the molecule. You aren't even testing the same drug anymore.
Exactly. It completely poisons the data set. A single atom out of place means the drug might fail entirely or become toxic.
Wow.
So the Harvest team had to build a custom parser from scratch to directly read the embedded ChemDraw binary files, the CDX files, bypassing the standard conversion entirely.
That is wild.
It seems like a tiny technical detail, but in computational biology, it is the difference between groundbreaking data and total garbage.
I get the mechanics of the agents, but let me push back on this multi-agent architecture for a second.
Sure.
Think about a game of automated telephone. If you have 5 sequential agents passing data down a line, doesn't that inherently increase the risk of cascading errors?
It's a valid concern.
Like if agent 1 hallucinates a target, doesn't that fundamentally poison the well for agents 2, 3, 4, and 5? It just seems incredibly fragile.
The researchers anticipated that exact vulnerability, and they countered it mathematically. First, by narrowing the semantic scope of each LLM call, the model isn't juggling multiple concepts, which inherently drops the hallucination rate near zero.
Because it's only focused on one tiny thing.
Right. But more importantly, they hard-coded strict guardrails. They set the LLM's temperature parameter to absolute zero.
Meaning the model is mathematically stripped of its creativity. It will return the exact same highly probable answer every single time.
Exactly. And beyond just setting the temperature to zero, they enforced strict grounding constraints.
What does that mean in practice?
When Agent 3 maps a chemical identifier, it is not allowed to generate novel text. It operates essentially like a strict copy-paste function. The system code requires the agent to find an exact verbatim text match in the original document to pull the identifier.
Oh, I see.
If the string is not physically on the page, the agent is mathematically forbidden from inferring it. This rigid constraint mechanism keeps the pipeline remarkably clean.
Which brings us to the ultimate question. We understand how the assembly line works, and we know the guardrails are in place. But does the output of this machine actually survive contact with reality?
Right. Does it work?
Does it match the quality of human experts? Let's talk about the results, because the scale they achieved here is genuinely hard to wrap your head around.
It really is.
The Harvest pipeline processed 164,877 patents in parallel running 50 full documents simultaneously. It extracted 3.36 million activity records.
Massive numbers.
It recovered over 365,000 unique molecular scaffolds and over 1,000 protein targets that were entirely absent from BindingDB. And the total cost for the cloud computing power to run all of this? $18,000.
Wow.
That breaks down to roughly 11 cents per document.
To put that scale into perspective, the human curation team at BindingDB takes about 2 full years to process just 1,500 patents.
2 years for 1,500.
Yeah. Harvest executed the equivalent of 55 years of human labor in under a week.
That's insane.
But as we established, speed and scale mean absolutely nothing without accuracy. So the researchers set up a benchmark. They took Harvest's automated extractions and compared them directly against the identical records that had been manually curated by humans in BindingDB.
Okay, head-to-head.
On those matched records, the automated extraction achieved a 91% agreement with human curators.
Wait, I have to stop you there.
Yeah.
91% sounds like a solid A- in high school. But in pharmaceuticals, an A- can kill people.
True.
If I'm building a billion-dollar drug discovery model, a 9% failure rate on raw data is terrifying. Why isn't it 100%? Who is making the mistakes?
That is exactly the right question to ask, and the answer is hidden in the residual analysis. When the researchers plotted out the numerical differences between the AI's extracted values and the human curated values, they didn't find a random spread of error.
How did they find?
They found highly isolated spikes at a delta of exactly plus or minus 3.
Plus or minus 3. What does that specifically indicate in chemical measurements?
Well, because binding affinities are measured on a logarithmic scale, a difference of exactly 3 log units represents a massive 1,000-fold error.
Wow.
It means someone looked at a table and confused nanomolar measurements with micromolar measurements. This is a classic, notorious unit conversion error.
Because human curators are staring at spreadsheets of microscopic numbers for 10 hours a day, their eyes glaze over, and they accidentally drop 3 zeros.
Exactly. So the researchers had to break the tie. They went manually back to the original source text of the patents to see who was actually right. The multi-agent AI pipeline or the human experts.
Yeah.
And they discovered that in these massive discrepancy cases, the AI agents were actually correct 92% of the time. The human BindingDB curators were correct only 5% of the time.
So the AI isn't just exponentially faster and cheaper. It is actively immune to the most common fatigue-induced human data entry errors.
Immune to fatigue, yes.
So what does this all mean for you, listening to this? It means the fundamental economics of scientific research just flipped upside down. Previously, to get a structured data set of this size, purity, and quality, you had to pay for a massive, heavily gated commercial subscription that only mega corporations could afford. Now, a small academic lab can generate a proprietary scale data set from public documents for the cost of a few months of a Netflix subscription.
It completely democratizes drug discovery data. But beyond shifting the economics, this new mountain of pristine data solves the exact problem facing modern AI that we mentioned at the start.
The generalization crisis.
Exactly.
Right, because Harvest didn't just extract data and put it in a folder. the researchers immediately used that newly liberated data to create something called 𝗏𝗏bench. What exactly is 𝗏𝗏bench?
So 𝗏𝗏bench is a rigorously constructed benchmark dataset derived entirely from the novel Harvest data. This means every single chemical record in 𝗏𝗏bench is guaranteed to be completely absent from all existing public repositories.
Why does that matter?
Having a secret, unreleased dataset is absolutely crucial for evaluating the real-world capabilities of modern state-of-the-art AI models like AlphaFold 3 or Boltz 2.
Because those models are essentially trained by reading the entire internet. They've already seen all the existing public data.
Right. If an AI is trained on all the molecules in BindingDB, and you then test its predictive power by asking it to evaluate molecules from BindingDB, you learn nothing.
Because it already knows the answers.
Exactly. You have no idea if the AI has actually learned the universal underlying physics of molecular binding, or if it just perfectly memorized the training set.
It's like an AI trained to paint beautiful, hyper-realistic human portraits. It can draw a perfect face looking straight ahead because it's seen a million photos of that.
Right.
But because it doesn't actually understand human anatomy, bone structure, or physics, the moment you prompt it to paint a person looking over their shoulder, it draws the neck bending backward at a broken 180-degree angle.
Yes.
It learned the visual pattern, but it completely lacks the physical rules.
That is a perfect analogy. To figure out if our drug discovery models actually understand the anatomy of chemistry, we have to give them a truly blind test. And to prevent the AI from cheating, the researchers used an integer linear program, an ILP. Think of an ILP as a mathematical force field.
Okay.
It guaranteed that the molecules selected for 𝗏𝗏bench were structurally separated and completely alien compared to anything the AI had ever seen in its training data.
So they quarantined the data to create a perfect leak-proof test environment. And then they ran Boltz 2, which is currently the leading open-source structure-based model, through this gauntlet.
They did.
What happened when Boltz 2 had to rely on physics instead of its memory?
The benchmark exposed a massive two-dimensional gap in the AI's capabilities.
Oh, no.
Yeah. Boltz 2's predictive performance degraded significantly when it was faced with novel chemical scaffolds, meaning it was handed molecules shaped fundamentally differently than its training data.
So it struggled with new shapes.
Yes, but its performance also degraded heavily when faced with novel, uncharacterized protein targets. When the researchers combined the two, asking the model to predict how a brand new chemical would bind to a brand new protein, the model's performance dropped terrifyingly close to random guessing.
Which raises a really uncomfortable realization. If these multi-billion-dollar AI models are just relying on structural familiarity and memorization rather than transferable binding physics, aren't we just building incredibly expensive pattern-matching machines instead of actual scientific discovery engines?
It's a very real concern.
I mean, if a novel virus emerges tomorrow and we point our best AI at it, it sounds like it might just hit a brick wall.
If we connect this to the bigger picture, this is exactly why the creation of 𝗏𝗏bench is so vital to the future of medicine.
Right.
It finally provides the rigorous, leakage-free stress test the industry desperately needs. By exposing exactly where these models abandon physics and fall back on statistical pattern matching, developers are now mathematically forced to build architectures that genuinely understand molecular interactions from the ground up.
Which is the only path we have to true de novo drug discovery for novel diseases that don't look like anything we've seen before.
Exactly.
Now, while Harvest is clearly a massive architectural leap forward, it's not magic. It relies on text and rigid parameters. And to give you the complete picture of this research, we have to talk about the pipeline's current blind spots.
Always important to do.
What can Harvest physically not do yet?
Well, there are a few key limitations in its current form. First, it completely struggles with what chemists call Marcouche structures.
Marcouche structures.
Yeah. These are highly complex combinatorial diagrams used in patents to legally represent huge families of molecules all at once using variable parts.
Oh, I see.
Automatically unpacking those diagrams into individual discrete compounds requires a level of spatial and chemical reasoning that text-based agents simply don't have yet.
It's like trying to read a recipe where the ingredient list just casually says, choose one of these 50 vegetables, one of these 50 proteins, and one of these 50 spices, and expecting the AI to instantly catalog all 125,000 possible distinct meals that could result from that.
That's a great way to put it.
It's an explosion of combinatorial possibilities.
Exactly that. Second, the pipeline cannot read graphical dose-response curves. If a scientist plotted their assay results on a visual line chart rather than typing the numbers into a table, the AI can't extract the parameters, it's just blind to the graph.
Gotcha.
And third, it struggles heavily to map ambiguous multi-subunit complexes. If a patent casually refers to a massive protein complex without specifying the exact subunits or domains being targeted, canonical mapping to a single uniprot identifier is incredibly difficult.
But there was one limitation mentioned in the paper that I found deeply, darkly ironic.
Oh, the safety policies.
Yes. The researchers noted that the LLM's own internal safety policies occasionally caused the system to completely refuse to process certain patents. Specifically, it blocked patents targeting high-risk pathogens, like the Ebola virus.
Yeah, that was wild.
The AI essentially viewed extracting public bioactivity data as a violation of its safety guidelines regarding bioweapons.
It basically triggers the commercial model's biosecurity tripwires.
Which is a fascinating friction point. Here you have a system specifically designed to accelerate the discovery of life-saving cures for deadly diseases, and it's actively refusing to process the data for Ebola because its own internal safety guardrails think the researchers are trying to do something malicious.
It perfectly highlights the tension between general-purpose LLM safety training and highly specialized scientific application.
Absolutely.
There is also one more structural limitation worth noting for anyone looking to replicate this. This entire pipeline, the flawless extraction, the near-zero error rates, the insane speed, only work because the U.S. Patent Office provides their documents as highly structured XML data.
Right, they aren't just uploading raw, scanned PDFs.
Exactly. If the researchers had been forced to use OCR optical character recognition to scrape the text off of raw image PDFs, the error rates and the chemical tables would have been catastrophic.
Because the AI wouldn't know where the rows and columns were.
Right. The underlying XML structure is what mathematically allowed the AI agents to confidently navigate the document hierarchy and isolate the tables. It underscores how critical it is for government agencies globally to maintain public data in inherently machine-readable formats.
So to bring this massive journey all together, what we've seen today is an absolute masterclass in how decomposing an overwhelming language model task into a specialized, rigidly constrained multi-agent assembly line can yield miracles.
It really is a paradigm shift.
Harvest rescued 55 years of dark data in a single week. It proved that properly constrained AI can actively bypass the fatigue and unit conversion errors that plague human experts. And by generating the pristine H-bench dataset, it created a mirror that finally forced the AI drug discovery field to look at its own flaws, proving we desperately need models that understand the physics of reality, not just the patterns of the past.
This raises an important question to leave on, actually.
Let's hear it.
If an 11-cent multi-agent system can instantly map the entire historical pharmaceutical landscape from dense patent texts, what happens when this exact same architecture is turned loose on disorganized clinical trial notes, or global regulatory filings, or decades of historical engineering archives?
Oh, wow.
We are rapidly approaching a reality where all of human expert knowledge, no matter how deeply buried or fragmented in unstructured text, becomes computationally active overnight. Who will control that newly awakened intelligence?
That is the multi-billion-dollar question. We started today by imagining a library where the books were written in invisible ink and scattered on the floor. Harvest didn't just find the blacklight, it organized the shelves, wrote a perfect unified index, and handed it back to the world. Thank you for joining us on this deep dive. Stay curious, keep exploring, and we'll see you next time.